Welcome back to the deep dive. Today, we are wrestling with, well, one of the biggest balancing acts in biology. This constant tension between keeping DNA the same, genetic fidelity, and the need for change the very stuff of evolution.
It really is a fundamental conflict. And you see the stakes immediately when things go wrong. Take Xeroderma pigmentosum, XP. It's a devastating inherited disorder.
Yeah, the sources kick off with XP. It's autosomal recessive, incredibly rare, but the effect is dramatic. Extreme sensitivity to sunlight. These kids basically can't go outside without protection. What's happening at the molecular level there?
It's a failure of the cleanup crew. Their skin cells are missing key DNA repair enzymes, specifically the ones that fix damage caused by UV light. So every time they're exposed to sun, the DNA damage just piles up, leading very quickly to skin cancers. It's stark proof of how vital these repair systems are. Life on Earth wouldn't work without them.
Okay, that really sets the scene. So our mission for this deep dive is to unpack this whole area. We'll look at mutations first, where change comes from, then the repair mechanisms that fight back. We'll also touch on some cool genetic tools like the complementation test and finish up with recombination, how variation gets shuffled.
Right. Let's start with the basics. Mutation. It's simply a heritable change in the genetic material. That's the definition.
And the first big distinction is where it happens, right? Somatic versus germline.
Exactly. Somatic mutations occur in your regular body cells. They only affect that individual organism. The classic textbook examples are things like the delicious apple or the navel orange.
Ah, right. Those started as a single mutant branch on a tree.
Precisely. And they have to be propagated vegetatively through grafting or budding. You can't pass them on through seeds because the mutation isn't in the germ cells.
So for evolution, for passing traits down in generations, it needs to be a germinal mutation.
Correct. That happens in the cells that produce gametes, sperm, or eggs, or their precursors. Those can be transmitted. If the mutation is dominant, you see the effect in the next generation straight away. If it's recessive, it might hide for a while until you get two copies together.
We also talk about spontaneous versus induced mutations. Spontaneous ones just happen. Errors in replication, maybe?
Yeah, often seemingly random background errors. Induced mutations are caused by specific external agents, mutagens, like radiation, or certain chemical.
And one thing the source highlights is that most mutations we can actually see phenotypically tend to be detrimental and recessive. Why is that the default?
Think about it. Genes and the proteins they code for have been optimized by millions of years of evolution. A random change is far more likely to break something or make it less efficient than it is to improve it, like throwing a wrench in a finely tuned machine.
Okay, that makes sense for detrimental. But why recessive?
Because usually, having one good copy of the gene, the wild-type allele, is enough. If you're heterozygous, that one functional allele often produces enough normal protein for the cell to get by. The functional copy compensates to the broken one.
Right, the cell can manage. So let's get down to the molecular nitty-gritty. How do these spontaneous changes actually occur? Watson and Crick had ideas about this early on.
A key mechanism is tautomeric shifts. The DNA bases normally exist in stable forms, like the keto form for guanine and thymine. But very rarely, they can temporarily shift into a less stable enol or imino form.
And if that happens right when the DNA is being copied?
Bingo. If the replication machinery encounters a base in its rare tautomeric form, it causes a mismatch. And that leads to a base pair substitution after the next round of replication. We split those into two types, transitions.
Right, where a purine replaces another purine, so A becomes G or G becomes A. Or a pyrimidine replaces another pyrimidine, T to C or C to T. 4 possibilities there.
And the other type, transversions. That's when a purine gets swapped for a pyrimidine, or vice versa. 8 possibilities there. Interestingly, transitions are generally much more common spontaneously. Probably because swapping purine for purine maintains the overall width of the DNA helix better than a purine for a pyrimidine swap, which distorts it more.
Okay, besides swapping bases, we also get frameshift mutations. Those sound nasty.
They usually are. Frame shifts happen when you add or delete one, or just a few base pairs, within the coding sequence of a gene. Because the genetic code is read in groups of 3 bases, inserting or deleting anything other than a multiple of 3 completely scrambles the reading frame downstream.
So all the amino acids after that point are wrong.
Pretty much.
Yeah.
You usually end up with a completely non-functional protein, often truncated early, because a stop codon pops up in the new garbled frame.
Then there are transposons, jumping genes.
Yep, mobile DNA elements. If one of these transposons jumps into the middle of a functional gene, it usually disrupts it completely. Mendel's wrinkle P allele. That was actually caused by a transposon insertion.
No kidding. Okay, and one more mechanism, the really unsettling one. Expanding trinucleotide repeats.
Ah, yes. These are short, 3-nucleotide sequences like CAG or CGG, repeated over and over. Sometimes, during replication, errors can cause the number of these repeats to increase from one generation to the next.
And that expansion is linked to some serious diseases.
Absolutely.
Fragile X syndrome is caused by an expansion of CGG repeats. Huntington's disease involves CAG repeats. Generally, the more repeats, the earlier the onset and the more severe the disease becomes. It's a kind of molecular stutter that gets worse over time.
It's amazing how many ways DNA can naturally go wrong. But then we learned how to induce mutations. H.J. Muller's work with X-rays in the 1920s was groundbreaking, wasn't it?
Huge. He showed definitively that radiation could increase mutation rates in fruit flies, Drosophila. He even developed a clever technique, the CLB method, to easily screen for new X-linked lethal mutations.
How did that work again? Basically, he crossed irradiated males with females carrying a special X chromosome, CLB, that had an inversion to prevent recombination, a recessive lethal gene, and a dominant marker, bar eyes. If the irradiated male's X chromosome picked up a new lethal mutation, then in the F2 generation, none of his grandsons inheriting that X would survive. So you'd see an absence of non-bar-eyed males. It was an efficient way to detect lethal mutations.
Clever. Now, looking at radiation types, there's a big difference between ionizing and nonionizing, right?
Yes. Ionizing radiation, like X-rays and gamma rays, has high energy. It penetrates tissues and knocks electrons off atoms, creating highly reactive ions and free radicals that damage DNA in various ways. A key point from Muller's work and later studies is the dose-response relationship.
You mean the single-hit kinetics?
Exactly. The frequency of mutations is directly proportional to the radiation dose. This implies there's effectively no safe threshold. Any amount increases risk.
Whereas non-ionizing radiation, like UV light from the sun,
That's lower energy. It doesn't ionize atoms, but excites them. It mostly affects surface layers, like skin. Its main damage mechanism is causing adjacent pyrimidine bases, especially thymines, to link together covalently, forming pyrimidine dimers.
thymine dimers, and those distort the DNA helix.
They do. They make it bulge and kink, which blocks DNA replication and transcription. And that brings us right back to Xeroderma Pigmentosum, where people can't repair these specific dimers.
Okay, let's switch to chemical mutagens, discovered during World War II with mustard gas.
Yes, Charlotte Auerbach found that mustard gas, an alkylating agent, was mutagenic. Since then, we've found many chemicals that damage DNA. We usually group them by how they work.
Like base analogs.
Right. These are molecules that look very similar to normal DNA bases, like 5-bromosyl looks like thymine. They get incorporated into DNA during replication, but they have wonky base pairing properties. They tend to mispair more often during subsequent replication rounds, leading mostly to transitions AC pairs flipping to GC pairs and back again.
And deaminating agents.
Things like nitrous acid. They chemically alter existing bases by removing amino groups. For example, it can change cytosine into uracil, or adenine into hypoxanthine. Uracil pairs like thymine, and hypoxanthine pairs like guanine. So again, this leads to transitions after replication. Works on non-replicating DNA too.
What about the ones that cause frame shifts?
Those are often acridine dyes, like proflavin. These flat molecules slip themselves, or intercalate, right between the stacked base pairs in the DNA helix.
Like wedging themselves in.
Exactly. This makes the helix more rigid and can confuse the replication machinery, causing it to slip and either add an extra base or skip one. Result, frameshift mutation.
This understanding of chemical mutagens led to a really important practical tool, the Ames test.
Yes, developed by Bruce Ames. It's a clever and relatively simple way to screen chemicals for potential mutagenicity, which often correlates strongly with carcinogenicity.
How does it work? You use special bacteria.
You use strains of Salmonella typhimurium that are auxotrophs. They have a mutation making them unable to synthesize the amino acid histidine. So they only grow if you provide histidine in their medium.
Okay.
You expose these histidine-requiring bacteria to the chemical you want to test, and then plate them on a medium lacking histidine. If the chemical is a mutagen, it will cause some bacteria to undergo a reversion mutation, changing the original histidine mutation back to a functional state.
So they become prototrophs again, able to make their own histidine.
Exactly. So if you see colonies growing on the histidine-lacking plate after exposure to the chemical, it indicates the chemical is mutagenic. The more colonies, the more potent the mutagen.
And the really crucial part is adding rat liver extract. Why do that?
Because many chemicals aren't directly mutagenic themselves. They only become dangerous after our own body's metabolic enzymes, particularly in the liver, process them. Think about nitrates in food or chemicals in cigarette smoke.
So the liver extract mimics what happens in a mammal.
Precisely. It contains those metabolic enzymes that can activate potential mutagens. Added it to the Ames test makes it much better at identifying chemicals that could be carcinogenic in humans, even if they aren't mutagenic to bacteria on their own. It's a vital safety screen.
Okay, shifting gears a bit. Let's say we have two different mutants with the same phenotype, like two flies with white eyes instead of red. How do we know if the mutations are in the same gene or different genes?
That's where the complementation test comes in. It's also sometimes called the cis-trans test. It's a fundamental genetic tool.
So how do you set it up? You need to get both mutations into the same organism, right?
Yes, you specifically need to create a heterozygote in the trans configuration. That means one chromosome carries the first mutation, say M1, and the wild-type allele for the second locus, M2+, while the homologous chromosome carries the wild-type allele for the first locus, M1+, and the second mutation, M2. So the genotype is M1 meto2 plus M1 plus MA2.
Okay, mutations are on opposite chromosomes. What does the outcome tell us?
It's beautifully logical. If this trans heterozygote shows the mutant phenotype, in our example, white eyes, then the mutations fail to complement. This means they must be alleles of the same gene. Why? Because the cell has a broken copy of gene A from one chromosome and another broken copy of gene A from the other. It has no functional gene A product.
Got it. No complementation means same gene.
But if the trans heterozygote shows the wild-type phenotype red eyes, then the mutations do complement. This tells you they're in different genes. The first chromosome provides a working copy of gene B, since M2+ is wild-type, and the second chromosome provides a working copy of gene A, since M1+ is wild-type. The cell has at least one functional copy of each necessary gene, so the pathway works, and you get the normal phenotype.
That's really elegant, like the example in the text with T4 phage mutants.
Exactly. Mutants affecting the phage head assembly fail to complement each other, showing they hit the same functional unit or gene. But a head mutant would complement a tail fiber mutant because head assembly and tail assembly involve different sets of genes.
Okay, so we've seen how DNA gets damaged and how we can analyze gene function. Let's loop back to defense. Given all these threats, life needs robust repair systems.
Absolutely, and there are multiple layers. One very direct mechanism, especially for UV damage, is light-dependent repair, or photoreactivation.
That uses a specific enzyme.
Yes, DNA photolase. This enzyme binds specifically to pyrimidine dimers, and when activated by blue light, it directly breaks the covalent bonds forming the dimer, restoring the original bases. Simple and direct.
But we mammals don't have that one, do we?
Strangely, no. Placental mammals seem to have lost it. So we rely heavily on other systems, primarily excision repair.
Excision repair, cutting things out. How does that work?
It's generally a three-step process. Recognize and cut out the damaged section, synthesize a new patch using the undamaged strand as a template, and then ligate or seal the patch into place.
And there are different types?
Yes. Base excision repair, BER, handles smaller problems, like removing a single damaged or incorrect base, such as uracil that arises from cytosine deamination. It leaves an AP site, a spot missing a base, which is then fixed.
But for bigger problems, like those bulky thymine dimers.
That's tackled by nucleotide excision repair, NER. This is a more complex pathway involving multiple proteins in humans. These include the XP proteins XPA through XPG.
Ah, the connection to Xeroderma Pigmentosum again?
Exactly. The NER machinery recognizes bulky distortions in the helix. An XP-chalesis complex makes cuts on both sides of the damage, removing a short oligonucleotide fragment containing the lesion, about 12 bases in E. coli, maybe 24 or 32 in humans. Then DNA polymerase fills the gap, and the EC seals it.
So a failure in NER is why XP patients are so sensitive to UV.
Precisely. It's our main defense against that kind of damage.
What about simple replication errors that the polymerase proofreading missed, like a G paired with a T?
That's the job of mismatch repair, MMR. This system scans newly replicated DNA for mispaired, but otherwise normal bases. The challenge is, how does it know which base is the wrong one in the pair?
Right. How does it know whether to fix the T or the G?
In bacteria like E. coli, it uses DNA methylation. The original template strand is already methylated at certain sequences, but the newly synthesized strand isn't methylated immediately. The MMR system recognizes the unmethylated strand as the new one, the one likely containing the error, and corrects the base on that strand.
Clever. And defects in human MMR genes cause problems too.
Yes, inherited defects in human MMR genes, which are homologs of bacterial genes like MUT-S and MUT-L, lead to a predisposition to certain cancers, most notably hereditary non-polyposis colon cancer, or Lynch syndrome. It highlights how crucial MMR is for genome stability.
Okay, what if the damage is so bad that the replication fork just completely stalls? Say it hits a dimer that hasn't been fixed yet.
The cell has ways to try and deal with that too. One is post-replication repair. It's a recombination-based mechanism. If polymerase stalls and leaves a gap opposite a lesion, the Regal-IK protein helps orchestrate a strand exchange with the sister chromatid to temporarily fill the gap, allowing replication to continue, hopefully buying time for the original lesion to be repaired later by NER.
But what if the damage is really widespread? Too many lesions?
Then the cell might trigger a last-ditch effort called the SOS response. This is induced by extensive DNA damage. It involves activating a whole suite of genes, including some for error-prone DNA polymerases, like DNA polymerase V in E. coli.
Error-prone. Why would the cell intentionally use a sloppy polymerase?
It's a desperate measure. The normal high-fidelity polymerase POL3 stalls at lesions. The SOS system basically says it's better to replicate past the damage, even if it means introducing a mutation, than to stop replicating altogether and die. POL3 can replicate across lesions that block POL3, but it tends to just guess and insert random bases opposite the damage.
So survival at the cost of increased mutation rate.
Exactly. It's a gamble, a trade-off between fidelity and survival when the DNA is heavily damaged.
Okay, one last major topic. DNA recombination. We mentioned it in repair, but it's also crucial for generating genetic diversity during meiosis. How does it happen at the molecular level?
The classic model is the Holiday model, named after Robin Holiday. It proposes that recombination starts when an endonuclease nicks single strands at corresponding positions on two homologous DNA molecules.
Okay, single strand breaks. Then what?
Then the free ends detach and invade the other DNA molecules, swapping places. This process, called single-strand assimilation, is helped by proteins like RK. The nicks are then sealed by ligase, creating an intermediate structure where the two DNA molecules are cross-linked.
The X-shaped thing.
That's the one, the Holiday junction, or Chi form. These have actually been visualized with electron microscopy. This junction can then move along the DNA, branch migration, extending the region of exchange strands. Finally, the junction is resolved by cutting and relegating the strands, which can result either in non-crossover or crossover products, depending on how it's cut.
And this process can sometimes lead to something called gene conversion. That sounds odd.
It is a bit counterintuitive. Normally, in meiosis, you expect alleles to segregate 2.2 in the products, like in fungal spores. Gene conversions is when you get non-reciprocal ratios, like 3.1 or even 5.3. One allele seems to have been converted into the other.
How does recombination cause that? It's not just swapping.
It's thought to arise from the repair of mismatches within the heteroduplex region formed during recombination. Remember, when strands exchange, you might temporarily have a region where one strand comes from one parent, say carrying allele A, and the other strand comes from the other parent carrying allele A. That's a mismatch.
So the mismatch repair system might kick in.
Exactly. The MMR system might detect this A mismatch in the heteroduplex. If it corrects the A to an A, using the A strand as template, then both strands now carry A. When this molecule replicates, it will lead to more A alleles than A alleles among the final products, hence the skewed ratio. It's basically recombination followed by mismatch repair acting on the intermediate.
Wow, so repair systems are involved everywhere, even in generating these unusual genetic outcomes.
They really are. It ties everything together, the initial damage, the multiple defense layers, and even the mechanisms that reshuffle the genetic deck.
Okay, let's try to pull this all together. We've seen mutations from tiny tautomeric shifts and substitutions to frame shifts and jumping genes as the source of raw variation.
Then we explored the defense forces. Sophisticated repair mechanisms like photoreactivation in some organisms, the crucial base and nucleotide excision repair pathways, mismatch repair for replication fidelity, and even the desperate SOS response.
And finally, recombination, explained by models like Holiday's, not only shuffles existing alleles, but through processes linked to repair like gene conversion, can itself subtly alter allele frequencies. It's this constant interplay, isn't it?
It absolutely is. Damage happens, repair pushes back, recombination reshuffles, and maybe, just maybe, there's a connection to something fundamental like aging. The text hints at this. If aging is partly due to the accumulation of somatic mutations over a lifetime, especially in long-lived cells like neurons,
then maybe the efficiency of our DNA repair systems is a key factor in determining lifespan. People with defective repair, like an XP, essentially show accelerated aging in some
It's
tissues.
a provocative thought, right? Could the limits of DNA repair be one of the ultimate limits on longevity? Something to mull over.
Definitely food for thought. Thank you for diving deep with us today into this intricate world of genetic stability and change. We really hope breaking down Chapter 13 like this helps you feel more confident with these core concepts.
And thank you from the Last Minute Lecture team.